Hello everyone, we are the Prion Research Peeps composed of Edane, Emily, Joe, and myself, Chris. The first questions that we must answer are, why do we care about prions? And what are prion proteins? To answer our first question, prion research is important because future research could facilitate breakthroughs in neurodegenerative disorders. To answer our second question, a normal cellular prion protein, PRPC for short, most simply is a protein that is most abundant in the brain. We are currently unaware of the physiological role prions play in humans. We do know that the misfolding of the protein in PRPSC causes brain damage and rapid decline in neurological functions. PRPSC can form protein aggregates, and those are called amyloids. Diseases caused by misfolded proteins are neurodegenerative and always result in death caused by coma or secondary infection due to loss of bodily functions. The most well-known prion disease is Creutzfeldt-Jakob disease, CDJ for short, which can be sporadic, genetic, or caused by the consumption of infected tissue. Other prion diseases include Gertzmann-Straussler-Scheinker syndrome, or GSS, fatal familial insomnia, or FFI, and PRP, cerebral amyloid angiopathy, or PRPCAA. These prion diseases are also known as transmissible spongiform encephalopathies, or TSE for short. There are other prion diseases that we'll flesh out more in later parts of the video, such as Kuru and BSE. All prion diseases are a form of amyloidosis, which is when the body builds up an abnormal amount of protein aggregates. CJD is primarily sporadic disease, but can come from transmission by ingestion or being exposed to PRPSC from contaminated bodily fluids. GSS and FFI are autosomal dominant diseases, which means that in one numbered chromosome, that is a non-sex chromosome, a single copy of the disease-associated mutation is enough to cause the disease. PRPCAA has been recognized as a genetic mutation in the PRP gene PRNP. So the human prion diseases include transmission, sporadic, and genetic forms. Structure of prions. We need gene expression to produce any kind of protein in our body. The prion gene is encoded in the short arm of human chromosome 20. The prion gene will be expressed into prions through DNA transcription and translation. Prion proteins have 4 levels of structure. The primary structure consists of only one sequence of peptides. It's the raw structure that has been translated. The secondary structures shown consist of alpha helices and beta sheets. These two structures are formed by hydrogen bonds. PRPC contains 3 alpha helix and 1 short beta sheet. In general, the entire prion protein has 230 residues. Here is what the prion protein looks like. Single molecules of PRPC and the subunits of aggregate PRPSC are tertiary structure proteins. The tertiary structure is a kind of 3D arrangement of secondary structure by water molecules or ions. Tertiary structure is stable due to its twists, bends, and hydrogen bonds. So that's why they are much more difficult to destroy than other pathogens. Meanwhile, the PRPSC protein cannot get into the cell as many other viral pathogens. But how can these proteins affect our body and result in neuron degradation? The protein can stick on the cell membrane through GPI anchors. GPI anchors will bind to the PRPC at the end of the sequence. Now here is the question. What does human PRPSC protein look like? The truth is we still do not know yet. The future research of prions might focus on this field. We do know the difference between the functions of normal PRPC and PRPSC and how PRPSCs become prions. PRPSC is the misfolding version of PRPC. Scientists use MD simulations to analyze the conformational change of the structure of proteins. In addition, X-ray fiber diffraction and cryon electron microscopy can be used to identify the protein's aggregation. The aggregation of PRPSCs is based on motifs. Several secondary structures can form motifs. Most typical motifs are beta hairpin and zinc finger. The motif structures are important in proteins because they always have specific functions. There is a unique motif structure in the amyloid, which is called cross-beta structure. Here is what a model of cross-beta motif looks like. in the het S prion, which is a prion of the filamentous fungus. Because of the fact that prion proteins are pretty similar in multiple different species of animals and fungus, the mechanism of aggregation in different species might be different, but the basic principle should be similar. The cross-beta structure is the basic pattern of the aggregation of PRPSC, and in this case, the aggregation of PRPSC will form the quaternary structure.
So, what causes PRPC proteins to misfold? Although the overall mechanism of prion misfolding is still largely unknown in the scientific community, some progress has been made in determining how specific mutations can destabilize PRPC's native conformation, thus leading to an eventual misfold. There are different ways for PRPC to misfold. In this section, we'll be talking about genetic predisposition to obtaining a misfolded prion protein, wherein a mutation in the PRNP gene causes the protein to destabilize and misfold. T183A is a specific mutation that causes the codon at position 183 to change from threonine, a polar amino acid, to alanine, a nonpolar amino acid. The secondary structure of the PRPC protein is determined by its primary structure, so this change would cause great instability. Authors of the paper, Mechanism of Misfolding of the Human Prion Protein Revealed by a Pathological Mutation, studied how the T183 mutation affected prion stability using circular dichromosome spectroscopy and differential scanning fluorimetry, DSF. CD spectroscopy uses a light source, filters, polarizers, and a detector to see how samples absorb circularly polarized light differently. This method allows researchers to determine the secondary structure of a protein. When authors compared the CD spectra of a wild-type, non-mutated PRPC protein to a mutant T183A protein, they found that both had a fairly similar secondary structure. However, the mutant version is slightly less alpha helical than the wild type, showing a slight change in conformation or stability. This finding was supported by the later thermodynamic tests of the CD samples and by using differential scanning fluorimetry. DSF measures the rate of protein unfolding as a function of temperature, so essentially, less stable proteins will denature at a lower temperature and more stable proteins will denature at a higher temperature. Both the CD melting curves and the DSF melting curves supported the author's hypothesis that the T183A mutation destabilizes the PRPC protein. This is shown by the T183A protein's lower melting curves in both of the methods used. Although not all methods used were mentioned in the video, they all came to one agreement. The T183A mutation causes the PRPC protein intermediate to become unstable, which greatly increases the likelihood of a normal PRPC to misfold to PRPSC.
Prion diseases have been known as transmissible since the early 20th century. The currently accepted protein-only transmission hypothesis states that the primary infectious agent of TSEs are simply the misfolded prion proteins. This is part of what sets them apart from the other pathogens, like bacteria, fungi, and viruses. The latter group all use a DNA template to propagate in the host body, while prion diseases are able to propagate from only the introduction of a misfolded protein. Possible pathways from ingestion to neuron infection have been proposed, but there are still many gaps in our understanding of the physiological routes prions take to form amyloids in the brain. After ingestion, the proteins may infiltrate the enteric nervous system through the intestinal mucosal cells, accumulate in the spleen or lymph nodes, and begin the aggregation process, and then travel through the spinal cord to the brain, likely mediated by myeloid dendritic cells. Most human TSE cases, however, are not caused by transmission. The most common human prion disease is CJD, usually caused by PRPC misfolding spontaneously, sometimes due to genetically heritable mutation. A small percentage of TSE cases are caused by the transmission of prions via ingestion or of exposure to infected tissues. Once the initial misfolding occurs, however, whether spontaneous or transmitted, it appears that a similar chain of events occur, resulting in the rapid accumulation of PRPSC amyloid fibrils in the brain and eventually death. The mechanisms by which normal PRPC are recruited and misfolded into pathogenic PRPSC amyloids are still being elucidated. though current research suggests that the N-terminus residues, 90 through 230, are critical in this conversion. In any case, there appears to be consistency in amyloid formation, whether the initial misfolding was spontaneous or transmitted. Once a prion amyloid is formed, it recruits and misfolds prion proteins more and more rapidly. High-rate conversions to amyloid form are essential to the propagation of PRPSC, because otherwise, enzymes called proteases would be able to differentiate PRPSC from PRPC and mark the misfolded protein for degradation. The amyloid structure inhibits proteases from being able to recognize PRPSC and allows them to escape degradation and continue to propagate. There are two well-known outbreaks of variant CJD caused by transmission between individuals. In the 1950s, a disease called Kuru was described in a remote population of New Guinea that had culturally significant cannibalistic rituals for their dead. The ingestion of their loved one's brain tissue, likely an individual with sporadic CJD, caused the spread of TSC within this population. It wasn't until 1996 that variant CJD was officially described in response to an outbreak of bovine spongiform encephalopathy, BSE, which you may know as mad cow disease. There is strong evidence indicating that variant CJD in humans can be caused by ingestion of beef contaminated with bovine proteins. BSE was first identified in UK cattle in 1986 with 2 cases. In 1995, there were 14,562 cases identified in farmed cattle. If prion diseases are so rare, how did this outbreak get so out of hand? The answer to this question drew global attention to the industrial practices taking place in cattle farms in the UK. The prion protein was able to spread so widely and rapidly because cattle were being fed beef products contaminated with prions. An uptick in CJD-like symptoms during this time led to epidemiologic and laboratory investigation that placed the developments of novel TSE cases within the incubation period of CJD, completely localized to the region of the BSE outbreak. This event is the most likely culprit responsible for the death of over 100 people from variant CJD in that region in the late 90s.
In conclusion, there still remains a lot to be done in the realm of prion disease research. The structure of human PRPSC still needs to be brought to light. We believe that this has not yet been done for a few reasons. Firstly, working with prions in a lab requires special precautions few people are willing to take for a disease with a 100% fatality rate. Secondly, due to the property of prion proteins, it has low transmissibility. Along with the awareness of avoiding consuming infraspecific tissue and increases in food safety regulations, human prion cases are rare to find currently. Though rare, there is still value in further investigation of these disease mechanisms because they bear resemblance to other neurodegenerative amyloidosis diseases. Future breakthroughs in understanding prion mechanisms can lead to development of treatments for amyloidosis diseases, of which there are currently none. For now, however, this insidious infectious agent continues to escape our understanding.